that. Okay.
So good afternoon.
Wait here. Okay.
Good afternoon. Welcome to the last section, biosensors. So let's start with the first speaker, Mario Di Acunto from CNR of Italy, with title of Plasmon Enhanced Raman Spectroscopy Inside Cells, What We Can Observe. Please.
Thank you. Secondo. Aspetta, dobbiamo primero condividere il schermo. Screen inside the program. Sorry, then just close this down.
Okay.
Okay, again.
Per la visione totale?
Where is the presentation?
Il PDF.
Home, okay, instrument. Okay. Where is it? I don't know. It's there.
Se no si...
It's here. Okay. Click there.
Yeah. Okay, sorry for the trouble. Thank you. Thank you, Chair. Good afternoon to everybody. And I wish to thank the organizer for the invitation and to task to bring this conference to conclusion in this difficult period. Okay, today I speak about the application of Raman spectroscopy combined with gold nanoparticles to perform spectroscopy inside cells and to give a description and a critical review on we can observe using such techniques. The basic ingredient is the plasmonic behavior of gold nanoparticles and the plasmon resonance and biosystem. And the Raman spectroscopy enhanced by the plasmonic behavior of nanoparticles inside the cells to perform intracellular spectroscopy. And I describe what we can do if it prevails the random factors or its possible reproducibility, and what of specific selection rules of a plasmon are enhanced and evidenced. And I performed the theory and experiment on osteosarcoma and osteodifferentiated mesenchymal stem cell. Okay, just 2 words on the plasmonic properties of metal nanoparticles. Generally, metal nanoparticles like gold, silver, or copper, when illuminated with electromagnetic wavelength, can generate an oscillation of electronic plasma inside, up and down inside the nanoparticles. And by changing dimension and the size and the shape of nanoparticles, spherical or nanorod, you can stimulate different modes. For example, between spherical or nanorod, or with nanoshell is a nanoparticle with the electric core narrow gold shell. You can tune the plasmonic resonance depending by illumination of your system. Just a word on Raman spectroscopy is a laser light scattering techniques. Practically, you excite a sample and collect the radiation of a vibrational mode of your system, and is strongly connected to polarization of molecules, so you can perform a fingerprint of different molecules inside your sample, and practically is very sensitive techniques to different molecules given by this effect. Is based on the discovery of Chandrasekhar Raman in the 1928. Is this discovery awarded with Nobel Prize a few years later. And practically we have that like the Raman scattering is elastic, it's elastic scattering effect. The cross section at this one depends about the Fart power of change of between frequency of the wave number of incident light and scattered, and this is also the reason of a blue sky due to scattering, relay scattering, and Raman scattering. Generally, you have an inelastic effect due to final red light. The final change is shorter with respect to phase absorption. To this is the intermediate state. And one problem is that Raman scattering is about 1 million times less intense to Rayleigh scattering, so it's very weak signal. This weak signal can be improved, can be enhanced using gold nanoparticles, and in particular dimers of gold nanoparticles. Inside the gap between the nanoparticles, so you have a great focalization of the field, and you can have enhancement of signal like this one. This is set signal to non-set signal. Generally, this is connected also to theoretical efforts to describe this effect. And for example, you can consider that the molecule inside this gap can be described by dipole signal connected to local electric field and polarization tensor. And the enhanced factor is can be described by this ratio between set signal and non-set signal, and N surf is the molecule scattering the set signal compared to volume nanoparticles. Generally, is 3 order of magnitude more intense CERTS effect. Okay, we applied CERTS, Raman spectroscopy and CERTS spectroscopy to different population of cells, changing from also differentiate the mesenchymal stem cell to cancer cell MG63 cell line. This is an experimental sketch of our system. It's a Raman spectrometer supplied by Thermo Fisher, and particularly is very improved by this collection part of the device, so having a very stable system. This is the parameter used for the experiment. We used green light, power changing from 8 to 10 milliwatt, and with time exposition changing 1/10 second depending by the experiment. Using Raman spectroscopy, the basic difference between also differentiated and cancer cell is represented in this plot. Practically, is very different rich landscape of biomolecules present in the cancer cell. For example, this is a Raman assignation. We have an increasing of the cysteine or isopatite with symmetric stretching of phosphate ion, or phenylalanine, amino acid, and other. And particularly, we looked for isopatite because in the Raman spectroscopy, the asymmetric stretching of phosphate ions output a particularly band of Raman signal at 960 centimeter means 1. And so performing Raman maps on the different cell, we have that, for example, looking at cysteine and heterospatite, we have a different distribution of such components in cancer cell with respect to osteodifferentiated mesenchymal cell. And this is generally red light is very high intensity of such components. Okay, what happens if we use CES? We introduce gold nanoparticles, in particular gold nanoshell, in our cell, and what we can observe inside the cell. Okay, we have nanoparticles rated high, 150 nanometers, with dimension less than 200 nanometers. The nanoparticle can be encapsulated inside the cell. This is the same image. This is the same image. This is bright field microscope. This is dark field microscope. And nanoparticles can be evidenced with high accuracy inside the cell. Okay, this is focus on a small region. This is inside the nucleus of the cell. And with dark field, you can enhance only the nanoparticle signal, and this is the equivalent Raman map. And we can observe that the different between the non-search signal with the search signal, we have a different band enhanced by the search effect with an increasing of about 2 or 3 order or degree. And now we focus our attention on the possibility of spectral, to solve the spectral mixture of what we are observing. What we are observing depends by the gap of the 2 nanoparticles, practically this region inside here. And this depends by the distance of the 2 nanoparticles, because the signal decreases exponentially with the increase of the gap between the 2 nanoparticles, depending by the polarization and the direction of electromagnetic field. And also theory was improved to create a search map. It's different between Raman map, because we enhanced only the search signal in our system. So now we have 3 different images. This is inside the cell with dark field. This is the Raman map. This is the search map. The red is, we are taking only signal in this region. And only in this region, we have 3 different signals. This is non-search, this is search, this is theoretically a curve solving the spectral mixture of biomolecules inside this region. And so we have the appearance of a new mode that experimentally are observed only a part. This is another sample. We have this image of a trimmer, but only 2 nanoparticles, the most close one each other nanoparticles, practically a dimmer, increase the search signal. So we have a signal practically only by dimers, practically. Okay, in conclusion, we perform Raman spectroscopy to discriminate with different cells, white cells and healthy cells and cancer cell. Applying search spectroscopy, working on the resolution lower than 1 micron, we can perform intracellular search spectroscopy. this is a very simple preparation of sample is very easy. You are practically depends what you are observing by the dimers distribution, the number of dimers, the relative gaps that you can control during the manufacturing of sample, and by improving the numerical approach on what you are observing, you can discriminate spectral mixture of inside the gap. In the next, the future perspective, we wish to improve the theory with ab initio molecular dynamics simulation. And okay, just to mention that this work was partially made in collaboration with Serena Danti, Alessandro and Luisa Trombi, and from Department of Medicine here at University of Pisa, and a special thanks to Thermo Fisher who supported this work. And part of this work was shared between several universities from Pisa, Rome, and collaboration with Japan. Okay, thank you for the attention.
So thank you so much for your nice presentation. Let's see if there is any question for you. So I think there is no question from the chat. I have a small question.
Okay.
It was very interesting, your method. When we have this kind of gold particles inside the cells, they should be like in a stable position to be able to measure the distance between them, or even if they have like...
There is stable position, yes. Also the signal is high reproducibility in the different days, for example. And the particular problem is that you have to use particular power, because you can risk to heat your nanoparticles, or destroying your cell, for example. In the first times, this was common experience illuminating the dimers, for example. And another interesting aspect is that the gold nanoshell absorbing the near infrared region, but when you have you can use green light, because you are stimulating resonance in the gap. So this is a sort of a third particles given by the particular shape of the dimers and the gap inside the two nanoparticles. So this is currently a random factor, limiting effectively the possibility to explore or to cell. You are at the moment basically depends by the dimers giving a certain effect, and so to explore the spot, this particular spot in your cell.
Okay, thank you so much. If there is no other question, thank you.
Okay.
So we can go to the next speaker, Valerio Poliani from IIT Italy, with the title of Plasmonic Ultra Small In Nano Theranostic In Out the Organism. Please, you can start.
Okay. Can you see my presentation?
Not yet.
Not yet. Oh, why? How can I do? Why I'm not?
Share your screen and then.
Okay. Try another time. Share screen.
And click on the screen that you see.
Okay.
Okay.
Now?
Yeah.
Oh,
now,
great.
Yeah.
Questo si può levare? No, aiuto. Okay. Is it possible to? Okay. Can you completely see my screen now? The presentation?
Yeah.
Okay, great. So, here I am. Thanks for the introduction and good afternoon. I'm Valerio Vogliani, a researcher of IIT, Istituto Italiano di Tecnologia. And in this talk, I'm going to discuss our approach for boosting the translation of plasmonic nanomaterials to the clinic. IIT, Istituto Italiano di Tecnologia, is a quite new institution in Italy, has its headquarters in Genoa, and there are several other centers in Italy. One of these centers is in Pisa, the Center for Nanotechnology Innovation, and this is the center in which I work. And here we have three main research lines that are devoted to the development and investigation of 2D materials to advance various kind of microscopies and the research line of nanomedicine. That's my research line. So the aims of my group are on one hand to shift noble metals to the bench side, to the bed side, and moreover we have some projects on the development of new nanocatalysts for energy and automotive. In the last year, we have done a lot of work on the side of bio research. And in this seminar, I decided to mainly give you a general overview regarding our research. For sure, if you are interested in some specific topics, don't hesitate to contact me or to follow us on socials. Well, why nanomaterials are so promising for medicine? Well, at the very first glance, for the sites. The sites is a common feature for all the nanomaterials, and has already resulted in the first nano revolution with the release of Doxil in the middle of 90s. The Doxil is a liposome loaded with doxorubicin, and it just brings more drug to the right side. There is another feature that is very interesting for nanomedicine, and that belongs only to metal nanoparticles, and is related to the light-matter interactions. This feature is the localized surface plasmon resonance, and by employing this kind of feature, it's possible to really start a new revolution for medicine by introducing new and efficient treatments, or also combined ones. Well, at the moment, where are all these promises of plasmonic nanomaterials? We worked, there is the literature for 20, 30 years. So where are all these promises? Well, if you are going to look at the market, you can directly see that there is no one plasmonic nanomaterial that is actually employed in clinics, and there are very few nanomaterials that plasmonic nanomaterials that are in clinical trials. This is related to the issue of persistence. What I would like to mean. The best, the plasmonic nanomaterials with the best theranostic actions are usually bigger than 20 nanometers. So after the action, they persist in the organisms, in particular in the excretory system organs, such as the liver, for example. As a first approach to increase the possibility to escape from the organisms, the size of the plasmonic nanomaterials was reduced to less to 5 nanometers. Why? Because with this size, it's possible to for these materials to escape from the renal pathway, so from urine. What's the problem here? That if the size is reduced, the plasmonic properties are lost. So there are no more theranostics action that can be employed. So what we have done for to bypass this problem, this dilemma? Well, we have developed a kind of nanoarchitecture in which a biodegradable capsule of silica contain inside a number of ultra small nanoparticles in order to obtain a similar plasmonic behavior such as bigger nanoparticles. In this way, we were able to combine the theranostic actions with the escaping from the body after the action. Here, just the production protocol that we employ and that we have standardized. I'm sure that Julia this morning has been enough clear in discussing this part. I would just like to recall that our procedure is really easy, scalable, and the particles that we produce are stable and intrinsically sterile, because produced and stored in ethanol. Now, a little bit of history behind the passion fruit-like nanoarchitectures. When I was, during my first postdoc in Spain, a lot of years ago, I was working on converting nanocrystals, and I was trying to modulate the emission bands of these crystals by introducing ultra-small gold nanoparticles in the structure. Well, every time that I did the synthesis, I obtained some strange byproducts. By the way, at a certain point, I look at these byproducts and I understood the potentiality of this kind of structure. So I then started to optimize the synthesis in order to obtain the standard protocol that we are now employing for the production of the nanoarchitectures. For sure, there is a subtle balance of ingredients. Never forget the stirring and the temperature. And that's right, in a couple of years from the waste byproduct, we arrived to the final nanoarchitecture structure. So from that time, now we are able to produce the particles with a lot of different metal inside, gold, platinum, silver, etc. And this is really interesting under the point of view that with the same protocol, we are able to produce different kind of particles, so a family of particles, in order to approach with the best particle, the right application. I would like now to introduce you the very first feature that I love of this particle, that is the degradation. I mean, the capsule, the silica capsule of our particles degrade in less than 1 day in full serum, but also in blood or and also in other physiological fluids. They are not only degradable, they are biodegradable because they also biodegrade inside cells. We performed investigations in several cell lines, and we always observed the fact that the capsule is degraded, and then the ultra small noble metal particles do not aggregate, but still remain single, so they are still able, after the biodegradation, to go out from the body. This is happened thanks to the glutathione concentration inside cells that is usually at millimolar concentration. Well, the other step that we did some years ago was to to assess the biocompatible of the nanoarchitecture, the very biosafety. This because we would like to care, not to kill potential pathogens. So what we observed was that, and what is really important here, is that at clinical concentration, at therapeutical concentration of nanoarchitectures, we obtained at maximum around 5% of malformation in zebrafish. This is really important because for the EMA, a therapeutic is not toxic if the malformation caused in zebrafish are under 15%. So we were very happy about this data. And now let's speak on a very pivotal topic about the nanoarchitectures. So the biokinetics, so the investigation about the biodistribution of our particles and their building blocks and the excretion. In order to investigate this behavior, we employed healthy mice, and we injected the mice with a therapeutical amount of nanoarchitecture. Then we collected every single day urine and feces, and at day 2, 6, and 10, so at 3 time points, we collected all the organs of the groups. Well, what we have observed at the very beginning, at day 2, the main amount of gold was in the liver. This is not surprising because we employed healthy mice, so without a target, the particles accumulate mainly in the liver. But during the days, what happens? The amount of gold go down until almost zero. And where this gold went? This gold went in the excretions that increased during the 10 days experiment. And we also observed that the main pathway, the main excretion pathway for the ultra small gold nanoparticles were in the urine, so they pass through the kidneys. In order to recall the biosafety of the nanoarchitectures and their building blocks, we observed that there was no toxicity for liver, nor kidney, nor other organs. So this data recall the zebrafish ones, a few slides ago. We then investigated what happens in the same models if we reduce the size of the metal nanoparticles inside the nanoarchitectures, and what happens by changing the metal, the noble metal inside. So by employing the same setup, we observed, as expected, that decreasing the size of the nanoparticles of gold, for example, here, the excretion increase, and that's expected. We observed that also silver nanoparticles are able to be excreted, but for platinum there is no excretion. And so from this data, what we observed is that gold, silver are both excretable but not platinum. Moreover, gold is always excretable by the renal pathway, but silver follow another pathway. Silver is excreted by mice by feces, so there is differences in the excretion pathway. Moreover, also here we did biosafety investigations on zebrafish, and we confirmed another time the non-toxicity of our nanoarchitectures. Very recently, we have investigated another type of administration, another pathway of administration of our nanoarchitectures, and we analyzed the inhalation pathway. This is in order to then study how these particles can act for oral cancers or lung cancer, etc. It's quite obviously, it's better to go through the inhalation pathway than by injection pathway for this kind of application. Also in this case, we employed healthy mice, and we collected excretions, organs at time points. And what we observed is that, for first, our particles by inhalation are able to join the lungs, so they do not remain in the upper respiratory tract, but go down in the lungs. This time we observe a very low quantity of gold in the liver. And as expected, our particles are able then to translocate from the lung to secondary organs and then to go out from the body. Here there is the analysis regarding urine and feces, and well, what we have observed here is that in the first 2 days there were a bigger amount of gold, and this is related. Day 1 and day 2, this amount is related to the ingested particles, because when inhaled, some particles are ingested. And then we observed a boost of excretion here by feces that is related to the particles that are translocated from the lung to secondary organs and then out from the mice. Another very important point to take into account here is that we observed a significant amount of gold in the brain. So probably during inhalation, there is a translocation of the particles from the nasal pathway to the brain. And more importantly, we observed that the gold in the brain then go down. So our particles are able to accumulate there in the brain, but then they are able to escape from the brain. Now, very briefly, because I think I'm almost finished my time, some insights about the passion-fruit light nanoarchitectures. So we employed our platforms for drug delivery. We inserted non-made cis-platinum prodrug, an octaedical complex that is released only inside cells thanks to the glutathione concentration in cells. So we have developed a double-controlled releasing system. The double control there is resulting from the degradation of the shell and then the interaction with the glutathione. So cis-platinum is released only in the target. And we have tested our nanoarchitectures in the 3D models that we have developed here inside my group. Then we have inserted also dyes inside the cavity of the nanoarchitectures, and we employed the quenching effect of the gold nanoparticles in order to compose some photoacoustic probes in the ER part of the spectra. Moreover, we were able to conjugate the photoacoustic behaviors with the ultrasound one. This is thanks to the impedance mismatch of the silica shell. So in this way, we obtained a light in, sound out, and sound in, sound out probe. Moreover, with a very small changing in the synthetic protocol, we were and we are able to produce some plasmonic bands here in the infrared part of the spectra. By radiating here on this band, our particles heat up. And very interestingly, after also many cycles of heating, the ultra small particles still remain ultra small, so they do not aggregate, for example, or reshape. So the excretion ability is still maintained. We tested our.
So you have 1 minute, please, because we should be on time. Can you please short your conversation?
Yeah. Just a couple of minutes more. So tested in 2D models and also in our 3D models. Here, some micrometastasis kind like cancer that we produce. So conclusion, we have produced the very first platform that is able to to combine plasmonic nanomaterials and the excretion. Now we are working in close collaboration with a couple of hospitals, and we are continuously develop novel 3D models of cancers. So I would like to thank all my group, all my collaborators, the former members of the group, and mainly my funding agency, l'Associazione Italiana per la Ricerca sul Cancro. And well, here are some bibliography. If you want, there is a special issue on nanomaterials in which I am guest editor. So if you would like to submit something, I'm more than happy. And thank you for your attention.
Thank you so much for your nice presentation. Let's see if there is any question for you. From the hall, there is no question and no question from the chat. So I thanks to you again. So we have to go to the next speaker, who is Jimena Alvarez Ferreira. I hope I pronounce it correct, with the title of Low Q Whispering Gallery Modes Optical Biosensor as Model Tumor Cell Surface for Cancer Immunotherapeutic Development. Please.
share your presentation, okay, you have to do it from the.
You can do it.
You did from here.
No, I have it.
This. And now you can.
PowerPoint, I think is good.
Yes, and this.
So, and is
Yeah, that one.
and that's it.
Yes.
Okay. Well, hello. My name is Jimena, and good evening to everyone. I'm here, a PhD student currently working in Surfly Nanotech, a startup located in Berlin. And I'm here today to talk a little bit about whispering gallery mode-based optical biosensors for the characterization and development of novel formulations of cancer immunotherapeutics. So first of all, what are whispering gallery modes? Well, whispering gallery modes are a specific type of waves or modes that were described for the first time in the St. Paul's Cathedral in London as waves that could travel along concave surfaces. They received the name of whispering gallery because when they were discovered because they observed that people could whisper against the wall of the gallery in St. Paul's Cathedral and be heard in the opposite side of the gallery. As it happened with sound waves, light can also produce whispering gallery modes when it is entrapped in spherical resonators, such as, for example, a microparticle. And this is the principle of optical-based sensors based on whispering gallery mode, as you can see in the scheme. So based on this physical phenomenon, Surfly, together with other partners, are trying to develop a new bioanalytical platform called WhisperSense for the label-free, rapid, and low-cost analysis of molecular interactions. From now until now, they have studied the interaction between DNA, proteins, or polymers. The current prototype is shown in the slide, and as you can see, it contains a fluidic chamber or fluidic chip with several or thousands number of wells. On these wells, the particles, which will be the sensor, will be placed, and the wells are small enough to make sure that only one particle fits per well. So the measurements are performed on individual sensors at each time. And how does it work? Well, the sensors have specific characteristics. One of them is that they contain a fluorescent dye, so they can be excited with a laser. After the excitation with a laser, some photons will start traveling along the particle surface, and due to the perfect spherical shape of the sensors, the whispering gallery modes will be created. And then these wavelengths can be monitored and detected with a high-resolution spectrophotometer and a detector. And how can we measure with this technology molecular interactions? Well, first of all, we need to measure this optical spectrum of the basal sensor. In this case, for example, we have a sensor with a ligand on the surface, an antibody. And we measure the optical spectrum. After that, we can add the analyte, which will bind to the ligand on the surface, and this will cause a change in the surrounding environment of the particle. And because of that, there will be a shift in the spectrum that is called mode shift. So there will be a shift from the red color to the red spectra to the yellow spectra. And this mode shift will allow us for the online detection and quantification of molecular interaction on the sensor surface. So the bigger this shift is, the bigger is the amount of molecules that are binding on the surface. And the whispering gallery mode technology can be applied to different fields and can have different applications, but today I'm here to discuss how this technology can be applied to the development or characterization of novel immunotherapeutics. Well, cancer immunotherapeutics are drugs or formulations that are used in cancer immunotherapy, which is a new or a recent type of therapy against cancer by boosting patients' immune system. And most of the immunotherapeutics are antibody-based formulations. due to their major impact in biomedical research and the clinical outcome. There are, the last year, there is increasing number of novel formulations and novel designs. And this has led to new challenges for the in vitro and bioanalytical characterization. And this is why we were trying to see if our whispering remote technology could be useful for this, to accept this challenge. So first of all, we need to optimize the production of the sensor. Sensors are produced from polystyrene particles of around 10 micrometers, which contain a fluorescent dye. These particles are coated up to 2 bilayers by the layer-by-layer technology with different polyelectrolytes. And we try to optimize this sensor production by checking or evaluating which of these three polymers, polyacrylic acid, polymetacrylic acid, or polystyrene sulfonic acid, comalic acid, is the best option for a coupled protein on the surface through the EDC-NHS activation procedure. So in order to do this, we coupled protein G, which is a really famous protein used in several biosensors and also in SPR, to mobilize antibodies or with the binding sites oriented to the media on surfaces. So what we did was to couple the protein G on the different polymers and to check the binding of the antibody to the sensor and the binding of the corresponding antigen. In this case, the blue bond. So as you can see on the right, we could detect the binding of the antibody to our sensor in red, and then we could detect the binding of the antigen to the antibody in blue. And when we analyze this interaction at different pHs and in the different polymers, we observed that polyacrylic acid in all the conditions gave out the best results. So from this data, our protein G microsensor were produced on polyacrylic acid L/L coating particles. And here, it's important to also measure the unspecific binding of the molecules to the sensor. And this is what is shown in this bar plot. When we added the antibody or the antigen to a microsensor coated with different proteins, not protein G, we could not detect any binding. So this means that the binding that I showed you before was really specific to the protein G and to the antibody. And also, we coupled fluorescent labeled protein, BSA in this case, just to see how was the distribution of the protein on the surface of the particle, because thousands of sensors are produced in one batch. So it's important that the distribution of the protein is uniform in between the different particles to have robust statistics. And as you can see in the images, the distribution is really uniform. And again, the polyacrylic acid gave out the highest in protein coupling on the surface. So from these proof of concept experiments, we moved to immobilization of therapeutic monoclonal antibodies. But instead of conforming ourselves with just adding the antibody and the antigen, we try to expand the microsensor to any kind of immunoassay, including those in which the antigen contains an FC tag. So in order to add an FC tag analyte or antigen to the sensor, we needed to first study or explore the saturation of protein G binding capacity by the antibody. And this is what we did. So here is an example of two different monoclonal antibodies, the tuximab and atezolizumab, in black and in pink. So when we added the antibody, we make sure that there is a saturation on the sensor. So this is, we make sure because we reach a plateau in the binding curve. After this plateau, we add a control and another IgG. In this case, is rituximab in red, and we get no binding. So this means that no protein G binding capacity was saturated. And then we add the analyte, and we detect a binding. So just to make sure, we performed several control experiments to make sure that the binding of this, the analyte that we detected here is specific to the ligand. As when we add other proteins or or no proteins at all on the surface of the sensor, there is no binding of the analyte. But, however, adding as much ligand as possible on the surface of a sensor is not the best option, because it can lead to aggregation or steric hindrance problems. So it's important to optimize the ligand density on the surface. So in order to do that, we reduce the amount of protein G on the surface to reduce the amount of ligand as well. And as you can see in the binding curves, while decreasing the protein G concentration, we could decrease the amount of ligand, in this case, cetuximab. But as you can see in this plot, while the amount of cetuximab was decreasing, the amount of the analyte remained stable, and this is in agreement with the idea that adding as much ligand as possible is not going to affect positively to the interaction. Apart from immobilizing the monoclonal or the therapeutic antibody on the surface, we try to do it in the other way around, to immobilize the antigen on the surface, and then so that it could be studied with the binding of the monoclonal antibody. And in this case, we try to immobilize several cell biomarkers in form of Fc fusion proteins. In this case, the idea, or the original idea, was to mimic somehow, and I say mimic because, of course, we are not recreating any cell, to mimic the cell surface of a cancer cell, so that we could add any cancer cell biomarker on the surface to study the development or the binding of new or novel therapeutic formulations. But again, as many of the current therapeutics, they are monoclonal antibodies, they again have an Fc fragment, so we need again to perform the saturation of the protein G binding capacity. And surprisingly, even though we perform the same protocols that with the antibodies, with the IgG antibodies, with the fusion proteins, we could not reach this saturation, because when we add the fusion protein, in this case in green, the EGFR, and we reach also the plateau on the binding curve. But then when we add the control IgG, we get still a really high binding. And this happened with all the fusion proteins that we tested, as you can see here in the bars. However, for the antibodies, we didn't see this. And this is surprising, because the fusion protein has the same Fc fragment as an IgG. So in order to check that this was not due to our setting, we performed the same experiments on SPR platform, and we got the same conclusion. The IgGs can saturate the protein G binding capacity, but fusion proteins cannot. And this is most probably due to steric hindrance problems in the protein G Fc fragment interaction. However, if we look at our data, we thought, okay, maybe we can use this high rituximab binding instead of such as limitation, such as an advantage. So we tried to block the free remaining protein G binding capacity with a rituximab before adding the analyte, so that we could measure the binding of the analyte to the corresponding ligand. But of course, we performed several negative control experiments, and in all cases, we found that the binding of the analyte that we detected before should be to the ligand. And just in case to check that the presence of rituximab on the sensor surface is not altering or interfering in the interaction, we tested two different binding pairs. So not two different binding pairs, one binding pair in which the ligand has not an Fc fragment. So we mobilized one cell surface biomarker in black, and then we added in one case rituximab, and in the other case we didn't. And then we flowed the analyte, which is a soluble ligand without any Fc fragment, and we compared the binding of the ligand in the two situations. And we observed the same amount of signal and the same behavior in the curve. So with this, we can conclude that at least in the experiments that we performed, rituximab was not altering the interaction. And to conclude, we have developed versatile protein G-coated optical biosensor that can be studied through whispering gallery mode in a label-free manner and can monitor antigen-antibody interactions of interest in cancer treatment. It's true that from now, or until now, we can only measure like qualitative binding steps. We cannot measure kinetics. We are working, developing, or improving the microfluidic system so that in the near future, we hope to be able to measure also KDs. Apart from antibodies, we have also immobilized cell surface biomarkers for mimicking somehow cell surface and characterize the binding of targeted monoclonal antibodies. This study has been performed with already existing monoclonal antibodies, but the idea is to apply these proof of concept experiments or these proof of concept sensors for validate the targeting of novel formulations. And this will be done in the future. And something that we found is that in contrast to normal to standard IgGs, Fc fusion proteins, even though they contain the same Fc fragment from IgG1, they cannot saturate the binding protein G binding capacity, and therefore we try to overcome this limitation by introducing a blocking step with an extra secondary IgG. And finally, I would like to thank Surfeit Nanotech, especially Dr. Lars, Kerstin, and Matthias, because due of their knowledge about whispering gallery mode. Also, the University Medical Center of Groningen, and especially Dr. Edwin Breimer, because for the providing all the immunotherapeutics and all the knowledge about immunotherapy, and also the other partners that form part of the iDirect project, and the organizers for inviting Surfeit today. And thank you. If you have any questions, yes, I will try to answer.
Okay, thank you so much. We see if there is any question. As usual, there's no question. So these are these results are from your PhD, yes?
Yeah.
Okay, good job. Thank you so much. So, go to the next speaker, Alexander Belushkin from Switzerland, with the title of Rapid and Digital Detection of Inflammatory Biomarkers Enabled by a Novel Portable Nanoplasmonic Imager. Thank you.
Hello, can you see my screen? Is the connection okay?
Can you hear us?
Yes, I can hear you.
We can't hear you. Just wait to.
You cannot hear. Can you hear me?
Okay.
Hello? Hello?
Yeah. Hello.
Okay, you can hear me now.
Yeah. You can start.
Okay, excellent. So, hello, everyone. My name is Alex. I'm from EPFL in Lausanne, Switzerland. So the topic of my talk is the nanoplasmonic imaging biosensor for digital detection of disease biomarkers. So just a brief outline of this talk. I will introduce you to the problem that we have tried to tackle of the unmet medical need in diagnostics. Then we will move to the core technology solution that we have developed at BIOS within my PhD. And then we will move on to the hospital results validation of this new technology that we got from a hospital in Spain. So imagine you go to a doctor and she decides that you need some medical testing. Now, depending, of course, where you are and the nature of the test, it may take you up to several days to get the doctor's appointment, for your blood to be withdrawn, transported to, large centralized lab where the samples are processed and the data is retrieved to the doctor. So again, depending where you are, may take you up to 3 to 5 days. Now, although for you it may just seem like an inconvenience of having to wait a couple of days, okay, it's not a problem. It's absolutely inappropriate for critical conditions like inflammation and other acute diseases, where it's really essential to have a rapid and cost-effective way to test directly on site, that a drop of blood can be taken from you, and within a couple of hours can be tested either at the emergency unit or at the doctor's office, with the immediate basically treatment directly on site. And one of such critical conditions is in fact sepsis. So sepsis affects over 30 million people every year worldwide, and it is the most expensive condition to treat in a hospital. This caused by body's own inflammatory response to an infection, such as, for example, bacterial infection that goes very acute. It goes very bad, very fast. In the US, it's the most expensive condition with over 24 billion in costs annually. And the problem with sepsis is that the chances of patient survival really drop drastically by 7.6% every hour that the treatment is delayed, which, as you may imagine, is a big problem when it compounds over time. And the problem is that the current ultimate microbiological diagnosis may take up to 72 hours to really get the microbiological decision on what the pathogen is. And this is why at the BIOS lab, we have been really trying to tackle this unmet medical need for rapid and affordable sepsis testing. And in fact, we have recently published the manuscript in small just this year, with the actual goal of being to tell, first of all, does the patient have an acute inflammation or not? And if there is an inflammation, is it of an infectious nature, in which case it's called sepsis and requires, antibiotic prescription to fight the bacteria, or whether it's so-called non-infectious systemic inflammatory response syndrome, or NISRS, as you can see here, which does not in fact require any antibiotics, just requires anti-inflammatory. And we have developed the test based on a novel nanoplasmonic mechanism that I will tell you about shortly. So the core technology is based on plasmonic gold nanohole arrays and gold nanoparticles, together with a portable reader device that includes basically a light-emitting diode, some chip cartridge and a holder, and a short optical path with a small objective and the camera. And the whole device, as you can see here, just a couple of, about 1 kilogram, few tens of centimeters long. It's very portable and low cost, and at the same time provides sensitive and rapid detection of disease biomarkers directly in patient samples without any pretreatment, with effectively allowing single nanoparticle detection. So this is how it works. So as I mentioned, the technology is based on so-called plasmonic gold nanohole arrays, which are essentially millions of nanoperforations in thin gold metal films that enable so-called extraordinary optical transmission, which is light transmission that is much larger than that predicted by classical diffraction theory. And so what happens is when we shine excitation light on the surface of the nanoholes, it excites so-called plasmons or electromagnetic oscillations in the gold film, and these oscillations that translate to a resonance peak in the far field. And because these resonances are highly sensitive to refractive index changes of the interface, we can observe it, as a resonance peak shift, just like an SPR in the previous talk. The classical use case of gold nanohole arrays has been, to detect in a label-free way. The problem with that, however, is when we want to detect very small molecules and in very low concentrations from complex samples, we first of all need a large and bulky spectrometer, and second, we're suffering from non-specific interactions from serum or plasma of the sample we want to detect. And that's why at the BIOS lab, we have come up with so-called nanoparticle enhanced plasmonic imaging approach that essentially enables digital detection of individual biomarkers and individual molecules. So what happens here is we have the plasmonic gold nanohole array here, and and we functionalize it with the first set of antibodies called capture antibodies specific against the antigen of interest. Then we'll take the gold nanoparticles and we functionalize them against the second antibody, which is called detection antibody. It's against the same antigen of interest, but a different epitope. So basically, the two antibodies can bind at the same time. And then what happens whenever we have the antigen in the solution, the nanoparticle can bind on the nanohull array surface. And when the binding happens, the gold nanoparticles disrupts locally the extraordinary optical transmission. And this results in the far field suppression of the transmission. So basically, we can see those individual small dark spots. And in this way, we can detect individual nanoparticles on the gold nanohull array surface. Now, I should just highlight here that, although both the nanoparticle and the nanohull are sub-wavelength in size, so the nanoholes are about 200 nanometers, the particles are around 100 nanometers, we can still resolve them in a conventional diffraction limited setup, basically a small microscope, because of those plasmonic enhancements. So it's important also that the gold nanohull array chips are fabricated with a scalable wafer scale manufacturing that uses deep UV lithography and ion beam etching that enables to yield around 50 chips from a single 4-inch wafer. That's really critical for translating the technology to in-field applications. And the next step for us was to develop a multiplexed array. So basically, we want to detect several things at a time. For this, we use a non-contact piezoelectric spotter, where on every chip, we make an array of hundreds of single spots, so that when we perform the bioassay, we can detect different things at the same time. As you can see here in the bottom left, on the image, we have 3 basically spots with the antigen of interest and 2 or negative control, which are just non-specific mouse isotype IgGs. And we can zoom in into the plasmonic image. So we can see here those individual spikes, right? But the question still arises, like, are they really nanoparticles? And can we see all of the nanoparticles that bind, or maybe not all of them? And how does this work? So in order to do that, we took the plasmonic image and we benchmarked it to SEM imaging, so basically scanning electron micrographs of identical areas. And we counted the nanoparticles with the SEM and on the plasmonic image. And we compared them basically 1-to-1 over different areas. So you can see here areas 1 and 2. Basically, 1 is on the spot with the nanoparticles, where you have nanoparticles bound, and 2 is just like negative background control without any nanoparticles, just to check. So you can see basically here with the nanoparticles, we have 3 kind of bright spikes, which are also corresponding to nanoparticles that bind inside of the nanoholes, while at the background we basically have nothing. So we repeated that time and over again over different chips, a lot of different experiments, and in total we counted over 1,000 different nanoparticles. And what we can see is that about 40% of particles bind inside the nanoholes on the SEM. And these are, in fact, those we can see on the plasmonic image as well. At the same time, we can see some of the particles that are outside the nanohole but close to the rim. And this is due to actually the plasmonic interactions, which you can find more details about in the paper if you're really interested about the plasmonic nature of those effects. But overall, we can see about half of the nanoparticles that are either inside the holes or close to the rim. Sorry. So the next step was actually the development of the portable digital plasmonic reader, which is essentially comprised of an LED light-emitting diode, a sensor chip holder, and an optical path with a small camera connected to a laptop for now. The overall device is less than 1 kilo. It's fully made of off-the-shelf optical components from Tor Labs and Nikon. And it's very easy to use, where you basically stick the chip into the device and we take pictures. And we have the algorithm that recognizes the spots and quantifies the biomarkers of interest. The next step was actually to enable the in-field use with a single step bioassay we wanted to have it as simple as possible so that we can perform tests outside of the lab. So the way the assay works, we take the nanoparticles and we put them directly into the serum sample. And then this whole mixture is loaded on the chip where the nanoparticles can bind on the surface of the nanoholes. And then we stick it into the reader, and this is kind of readout typically that we can see. So we can see 3 spots with the antigen of interest and 2 negative controls. So we developed this assay for 2 different biomarkers. One is called procalcitonin, or PCT, which is a biomarker related to sepsis. The typical challenge with detecting it is that it's a very small molecule, small peptide, and it's typically present at very low concentrations, so difficult to detect. And the second is called C-reactive protein, or CRP, which is a more general biomarker of inflammatory response. It's a larger molecule and also present in higher concentrations, so fairly easy to detect. And just a quick illustration, I hope the video works. Yes, so you can see nanoparticle binds in the solution just by diffusion to the antigen of interest. Then as it diffuses to the surface, with time it binds to the antibodies functionalized. And if it's in the hole, we can see it basically as a transmission in the far field. And over the field of view of the image, we can count those individual particles, detect and quantify the antigen. So having that on hand, we packed all our stuff and embarked on a trip to Barcelona from Switzerland, not so far away, but still you have to take all the stuff to test with the real samples. And before doing the actual patient sample testing, we had to determine what's the sensitivity limit and the dynamic range for both PCT and CRP. And as you can see here, we have pretty similar performance. So for PCT, we have about 21 picograms per milliliter limit of detection and about 3 orders of magnitude dynamic range. And for LOD of CRP, it's about 36 picograms and very similar dynamic range. So next, having characterized the system, we established the tests with the patient samples. So we had 34 patients with sepsis, non-infectious SIRS, and healthy controls. In particular, we had 18 patients with sepsis, 11 SIRS, and 5 healthy. Most of the patients who had septic response, they were post-infections, basically acute respiratory or abdominal infections mostly. Unfortunately, not all of the patients were surviving. Again, highlighting the need for sepsis treatment and care. So what we did first, we quantified 2 biomarkers for the most typical patients in PCT and CRP for the 3 groups. And as you can see here on the right, for different level of the biomarker of interest, for different level of PCT, you have very different density of nanoparticles binding that allows to, quantitatively detect the level of biomarker. At the same time, we mapped the results of plasmonic signals, basically whatever we get with our system, to the gold standard CLIA, which is chemiluminescent immunoassay, just the tool that they use in the hospital for performing those tests on commercial machines. And we got pretty good agreement between both, so this was also very encouraging. So having that, we basically moved and quantified all of the 34 serum samples that we had for both PCT and the CRP. As you can already see here, PCT seems to be a more discriminative predictor between the 3 groups, while CRP is kind of non-specifically elevates for the inflammation, but it's very hard to tell whether it's infectious nature or not. At the same time, although the serum samples had pretty different optical properties, like different turbidity, different color, different consistency, the results were very consistent, again, showing the robustness of our system. So the next question for us was, okay, that's of course looks nice, but the ultimate, impact in the future is actually helping to diagnose, helping to predict the disease and compare that to laboratory immunoassay. So how good can we be in terms of assisting diagnosis. And in order to do that, we took the plasmonic signals that we obtained for each of the patients, and we mapped them one-to-one to the ultimate clinical diagnosis that we had for each of the patients. And we saw how good of a discriminator our plasmonic signals were. Of course, again, this was not to validate the biomarker. The patient cohort for biomarker validation here is too small. This was mostly to validate the technology. But in any case, we did the same for laboratory immunoassay, and we got very similar discrimination in between the three patient groups with the lab immunoassay signal and with our plasmonic signal, again showing, you know, the robustness of performance. So finally, sorry, I don't know why it's clicking through, like it's skipping one always, but anyway, the point was to show the rapid detection of biomarkers. So in order to do that, we can in fact perform live analysis. So basically take a video and we plot the signals in real time from the spot versus the control background. And then we can have the cutoff time where the signal exceeds the background and compare it for the sepsis, SIRS, and healthy groups. And in this way, we can see that septic people we can identify within 5 minutes. For SIRS, it takes a bit more, around 10 minutes. And then for healthy controls, basically, it's just the background signal. There is almost no biomarker. So the total time can be just below 15 minutes for the, you know, handling, insertion, and the detection, just, you know, for the whole assay. So on that, I would like to just to sum up. In this work, we basically show how, you know, powerful combination of nanophotonics, nanofabrication with surface chemistry techniques, and microfluidics can enable the next generation of biosensors that can actually be deployed in a portable, low-cost setup within actual hospital settings to provide new point-of-care diagnostic solutions. And on this, I would like to say a huge thank you to the whole BIOS lab, and especially Professor Altug for her support, and of course the support of different funding bodies, in particular Enable Grant of EPFL, and our amazing Center of Micro Nanotechnology that really helped a lot with the fabrication and the establishment of this nanotechnology. And of course, thank you very much for your attention.
So much, Alexander, for your nice presentation. Actually, what you and your group have done is very interesting. And let's see if there is any question from the hall and chat for you. Okay, I think there is no question. So thanks again. We can go to the last speaker, who is Bahar Azimi, because we lost Joanna in the first section in the morning. So she presented in the second section in the morning instead of me. So I am going to present now very fast, and then we will close the section. So I'm going to talk about the position of chitin nanofibril by electrospray technique on biocompatible biopolymeric surfaces. As you know, like fossil fuel-based products have a lot of application and usage in different industry in these days because of their cheap availability. But they are not biodegradable, and so they are not like environmental friendly. And actually, their disposal is one of the like serious issues to be considered in these days. So because of like serious environmental worries related to the fossil fuel-based products, the researchers are encouraged to replace these like petroleum-based products with sustainable natural friendly materials. So we can say that natural biopolymer-based products have received a lot of attention and they have found a lot of application in different areas, including cosmetics or skincare application. And the key specific properties of this kind of materials are biocompatibility and also biodegradation, both in the environment and also in the human body. We can say that chitin and lignin, byproducts of fishery and plant biomass, can convert to some high-value bio and eco-compatible products, and in nanoscale, we can improve their properties, including antibacterial properties, anti-inflammatory, and also anti-aging activity with controlling their crystalline structure and also their purity. Since this kind of natural biopolymers are very important, this is the reason that we are using them in a different project that we are working. For example, as you can see here, in these three European projects, Polybioskin for nano-eardrum and also ECOT, we are using this nanoscale chitin, nanofibrils, and also nanolignin to modify the different surfaces, different substrates. Let's start with Polybioskin project that also today morning Beatrice explained very well about the product that we obtained in this project. So as you can see here, to produce this chitin nanofibrils or nanolignins, or even complex of chitin nanofibrils with a nanolignin, we used spray dryer techniques. And as you can see here, with this technique, we could reach to the nanoscale of each material. As you can see here, we have chitin nanofibril. Here we have a nanolignin, and here you can see the complex of chitin nanofibril with nanolignin. To have better view about the morphology and their size, we diluted them in distilled water, and again we try to have some SEM image. And as you can see here, this is the structure of chitin nanofibrils that we obtained with spray dry technique. And also you can see here the complex of chitin nanolignin, including also glutaraldehyde acid. And you can see both structures that are are together. Then we investigated their thermal stability, and our results show that these bioactive complex are thermally stable, and they are stable around 130 degrees. Which is good for different application. And as you can see here, we investigated the chemical structure of our complex, and as it can be seen here, we could detect the glutaraldehyde acid in the structure of the spectrum of the chitin lignin complex. Then we investigate some biological characteristics of this material, and the results show that actually we did in vitro cultures of human keratin oxides and also human mesenchymal stem cells for potential use in skin contact applications. And results show that results show the good cell viability of these complexes, and also they are like cytocompatible because metabolic activity of all samples significantly increased over the time, as you can see from the results of Alamar Blue test. And also they show good anti-inflammatory activity, and they may serve for the delivery of biomolecules for skincare and regeneration. You can see here that mineral matrix deposition results also show that the both treated and untreated human mesenchymal stem cells cultured in osto differentiation medium actually produce mineral granules, and as you can see, like visible in black, and with no appropriate differences according to the morphology and extensions, that which shows like good biological activity of this complex. Also, all samples that in fact show could downregulated the expression of pro-inflammatory cytokines. And as you can see, the complex of chitin, nanofibrils, and nanolipin, including like glutardic acid, was the most effective in modulating the expression of the largest number of cytokines, especially IL-1 alpha and IL-6. And also, as you can see here, the complex of chitin nanolignin was able to upregulate the expression of an antimicrobial peptides, the human beta defensin, HBD, which is involved in innate immune response. If you are interested to know more about these results, you can check our paper that we published in this journal. And according to these results, we decided to... Sorry? So according to these results, we decided to use this kind of nanoscale natural biopolymers to functionalize the surface of different substrates. As I told you, for example, for polybio skin, we decided to functionalize the surface of the electrospun nanofibers, polyhydroxyalkanoids, and using electrospray technique. And as you know, this is one of the most important methods to produce nanofibers and nanoparticles. So with changing some parameters of electrospray technique, we reach to the different results. As you can see, for example, by using different solvents, we reach to the nanoparticle with different morphology and size. Actually, by using acetic acid, ethanol, and distilled water, we couldn't reach to the very separated particles. They were like aggregated. But by using acetic aqueous acetic acid, we get good results. We got good results. You can see the nanofibrils are very in a uniform shape and like nanoscale size. So we change by using the same solvents. We change the parameter of electrospray technique, and we use higher voltage, and also we decrease the distance between the tip of the needle and the collector. And in this way, in this new condition, we could successfully produce chitin nanofibrils even using ethanol as solvent. So this is it shows that how much the electrospinning parameters are important to optimize and reach to the desirable product. After we optimize the electrospinning and electrospray condition, also we try to electrospray chitin, lignin, acetic and glucardic acid to reach to the some nanoscale size. And as you can see, for in the case of complex, the size of particles are not very uniform. In some case, we have bigger or maybe aggregated like complex, but in some case they are very small, so it's not very uniform in size and shape. But in the case of chitin nanofibrils, we obtain very good and uniform size of particle. In the next step, we use in the optimum condition, we try to use electrospray technique to functionalize the electrospun fibers. We had polyhydroxyalkanoate as substrate, and we electrosprayed chitin and chitin lignin complex on the surface of this nanofibrous mesh. And as you can see, they could successfully located on the surface of the mesh, and they kept their size and their morphology. We investigated the chemical structure of the functionalized mesh, and as you can see, all of the main absorption bands of the chitin and chitin nanolignin can be detectable on the surface of the functionalized mesh without any change. So we investigate some biological properties of this substrate, and as you can see, the results show that they are promising materials to be able to use for some biomedical application, especially in contact with the skin. And here you can see the paper that we already published according to these results. And if you are interested, you can take a look on this paper. So as conclusion, I can say that these results are very encouraging for the use of this kind of natural biopolymer, bio-based like chitin nanofibrils or chitin nanofibrils complex with nanolignin, for especially for some skin contact applications like cosmetics or tissue repair. And electrospinning technique is a very interesting method that at the same time we are able to produce fibers and nanoparticles, and also we can use this technique to functionalize the different surface, including nanofibrous mesh. So at the end, I have to thank my supervisor, Professor Andrea Lazzeri, Dr. Serena Danti, Dr. Maria Beatrice Coltelli, and Professor Patrizia Cinelli, and all of my colleagues who helped me to reach to these results. And thank you for your attention. So I'm here if there is any question. So let's see if there is any question on chat. No. So I think Serena is going to come here to close the section. Maybe yeah, maybe we have to wait for them. Okay, she's here.
So thank you. We arrived to the end of this conference that was really exciting for me because I could really collect the different variegated but very significant speakers. So I think the topics are very impactful, and so I am very proud also of the work of my collaborators, Ugo, and I think this conference is very successful. So I really thank the people here and the people at home who are following, who spent their time these days following by the computer and not be here in person, unfortunately. So we really hope that the next edition of this conference will be held in person in Copenhagen, and we hope to see this such a beautiful city, and also to be in touch each other and see the progress of this work. I think I collected new speakers in this conference that were not before in the previous editions, so maybe they are keen to continue to follow this event. So I want to thank the University of Pisa for the great efforts, the huge work that they did in the backstage, from the direction. They are really the key people who made possible having this event in this way, so in person and in remote, with like such beautiful direction. So I think this is, they were wonderful, and also my students and the other people involved in the preparation. So I really thank everybody, and I wish to see you again, hopefully in person. So by this, I close officially this event, and you have the link. We are in touch. I will send you some emails in the next days. So the link are on YouTube, and you can visit again and see these sessions by remote. So thank you very much, and a big applause to all the people working for this event. Also a special thanks to Josephine, who was very helpful. Thank you very much. Thank you. Bye-bye. Okay.